Genetics and molecular biology show up constantly on the MCAT, but they don't have to be confusing. Today, we are diving deep into exactly what you need to be able to do great on these high-yield biology topics. We're going to cover DNA structure, replication, transcription, translation, gene regulation, Mendelian genetics, inheritance patterns, mutations, and biotech tools like PCR and gel electrophoresis. By the end, you will be fully prepared for MCAT questions in these areas. First, let's look at DNA structure and replication. You need to know DNA structure basics, that DNA is double helix with sugar phosphate backbone and bases A, T, G, and C. You also need to know complementary pairing. Adenine to thymine, which is with 2 hydrogen bonds, and guanine to cytosine, which is with 3 hydrogen bonds. You need to know anti-parallel strands, 5' to 3' direction and vice versa. You also need to know DNA replication process, semi-conservative replication, so each new molecule is one old strand and one new. You need to know the key enzymes. Helicase unwinds the double helix. DNA polymerase, it builds new DNA strands in the 5' to 3' direction. Primase, it places RNA primers. Ligase connects Okazaki fragments on lagging strands. And you need leading versus lagging strands, so the Okazaki fragments. Make sure that you know enzyme functions very clearly. The MCAT does often test enzyme roles. Let's move on to transcription and RNA processing. So transcription overview, DNA goes to mRNA in the nucleus, and then RNA polymerase synthesizes RNA from DNA template. Promoters and terminators signal start and stop. You need to know coding sense versus template, antisense strands. RNA matches coding strand, but with U instead of T. You need RNA processing, introns, so non-coding, removed exons, coding, and joined splicing. 5' cap added, so stability initiation is translation. Poly A tail on 3' end, stability export from nucleus. The MCAT frequently asks about mRNA processing, so don't skip the details here. Let's move to translation and protein synthesis. You need the translation basics, so it occurs in cytoplasm on ribosomes. mRNA, amino acid sequence, so proteins. tRNA matches mRNA codons with correct amino acids. Make sure you know the steps. Initiation, ribosome binds mRNA at start of codon, so AUG to methionine. Elongation, tRNA adds amino acids forming a peptide chain. Termination, stop codon, UAA, UGA, UAG, it ends translation. And then genetic code, codon, 3 base sequence, for example, AUG equals methionine. Codon table, be familiar, you don't have to memorize, but you want to make sure that you know the start and the stop codons. Watch for questions that are testing codon, anticodon pairing, and mutations. Let's move on to gene regulation. You need to know prokaryotic regulation, so operons, operon structure, promoter, operator, genes. You need to know lac operon, TRP operon, as well, so it's inhibited by tryptophan. You also need to know eukaryotic regulation, transcription factors, enhancers, and silencers. You need to know about epigenetics, DNA methylation, histone modification. So let's look at genetics and inheritance patterns. You need to know Mendel's laws, the law of segregation. Allele pairs separate into different gametes. You need to know the law of independent assortment. Separate genes sort independently. Know your Punnett squares. Monohybrid cross, 3:1 ratio. Dihybrid cross. Test crosses. Heterozygous versus homozygous. And know your inheritance patterns. MCAT loves pedigrees, so make sure that you practice identifying inheritance patterns very, very quickly. Let's move on to mutations and genetic disorders. You need to know point mutations, missense, nonsense, and silent. You need to know frame shift mutations. You also need chromosomal mutations, duplications, deletions, inversions, translocations. You need to know the impact on protein function, know how mutations affect phenotype. Mutations tend to show up in experimental passages, so practice predicting outcomes. Let's move on to biotechnology techniques. You need to be familiar with PCR. It amplifies DNA segments. You also need to be familiar with gel electrophoresis. It separates DNA by size. Restriction enzymes cut DNA at specific sequences. And cloning inserts DNA into vectors. Understand the practical applications and principles behind each technique. So quick review: DNA structure, replication, transcription, translation, gene regulation, genetics, inheritance patterns, mutations, and biotechnology techniques are your highest yield MCAT topics in genetics and in molecular biology. Make sure that you review these regularly. If this helped clarify genetics and molecular biology for the MCAT, hit that subscribe button for more MCAT deep dives, strategies, and study tips. Let me know what MCAT bio topics you'd like to see next. Thanks for watching and good luck studying.